everyone, very good afternoon. Okay, can you drop a thumbs up on the chat box so I know that all of you are here and I'm audible and all of you are awake. Drop something, say a hi, say good afternoon in the chat box so I know that all of you are awake. Oh, dekho, look who's here. We have Nivel here. After such a long time, Nivel. Am I audible? Everyone, can you drop a thumbs up or something on the chat box so that I just know that I'm audible to all of you? I'm audible, thank you. Upasana is the only one following instructions here. Bas, thank you Upasana. Upasana is the only one following instructions. I asked you kids to drop me a thumbs up and she's the only one who's done that. Saksham, don't be so hard on yourself. Take it easy. Hi Vaibhav. Hi everyone. Very good afternoon. Very good afternoon. Hi Pravesh. Okay, how many of you went through the PDF of the one-shots that we took for human health, for biotechnology, principles, and even for applications? How many of you went through that? And how were the sessions for one-shots? Are you ready for some more one-shots before our board exam, before your NEET exam? Are you guys ready for some more one-shots before your NEET exams? Very good. Yes, and yeah, we will be also covering Nivel, Shreya, Pravesh, Mirdula. We will also be covering organisms and population. This is a chapter which I did not teach you in our lives, in our online classes, but over here we will be covering that. Okay. Alright you guys, let's begin. This is going to be 2 days. Today and tomorrow we will be having 2 sample papers which we're going to be discussing. Yes. So I will be also just guiding you on how you're going to be attempting your CBSE paper. Remember, this is term 2. It is not going to be objective. It is a subjective paper. Unfortunately, right? हमको पसंद नहीं है मैम. We are so used to just ticking our answers. We don't like writing long sentences now. है ना? Shri, we're going to take most of the chapters. The calendar will be up very soon, just like how I had this week's calendar up. Like that, every week's calendar will be up very soon. Subjective is good. Even after this, all these 2 years of doing so many MCQs, hundreds and thousands of MCQs, Nivel still tells me that ma'am, subjective is good. Okay. Acha Saksham. Then Saksham, you're done with 12th standard. Then you will answer all the questions that I ask you. Right? Hi Anumita. Very good afternoon. Very good afternoon everyone. So yes, let's begin since most of you are here. And yes, today's session is going to be one sample paper which we will be discussing so that you know how to attempt your board exams, right? So yes, quickly everybody, since you're all here, go and press that little button that's called as the like button. Share this video with your friends so they can come to during the live session. And also I hope that you already have done this, but please go and subscribe to our channel so that we can keep delivering to you such important and fun topics right before your exams to stay motivated. Saksham, ritual of roasting you always will be there. Alright, so those of you who are new to my channel, to this channel, a very warm welcome. Let me introduce you to myself. I'm Dr. Sindur, NEET zoology expert at Vedantu. I have been mentoring kids for over 10 years for their NEET exams, and I have done my Bachelor's in Homeopathic Medicine from D.Y. Patil College in Pune. And so I have been teaching kids for cracking their NEET exams, and I am so thrilled to see all my long-term session kids also over here in the chat box. All of my long-term session kids, drop me a heart on the chat box. I want to see how many of you are here. All my long-term session kids, I want you to drop me a heart on the chat box. I want to see how many of you are here. Acha, Nibel has his reasons. Hi Nikhil. Hi Jaya, Mihika. Aja Saksham, aja. Come. We're waiting for you. Hi Pravesh. Very nice. Nibel. Hi Satyashri. Hello everyone. And how many of you all have obviously attempted your 10th standard board exams hopefully, and you have cleared it hopefully in a very proper legal way. I hope you have cleared your board exams, not with the help of your neighbor. So I'd like to know how many of you actually go ahead and read the instructions which are provided over there. Tell me, how many of you actually go and read the instructions? I never used to do this. Never. I never would read the instructions. I would never read the instructions. Yes, Mihika, ये वही सक्षम है. और कोई सक्षम है क्या? Shreya reads it. Nivel does not read. Pravesh does not read. Never. Henna, I used to be the same. And Shreya is one good girl we've got in this classroom right now. So, first of all, reading the instructions is very important, because many kids I know, they will be asking me, ma'am, are there options? Ma'am, do we have to attempt everything? Arey, you're giving the exams, go and research, read the instructions, do everything. You do religiously very good. Where am I from? India. Okay, so all the questions here, all the questions in your paper. First of all, tell me, how long is this paper going to be? 2 hours or 3 hours? How much time do you have for this paper? 2 hours or 3 hours? Tell me. Acha Saksham. Very good. Everybody knows that you have 2 hours. Acha, how was your physics paper, by the way? How was your physics paper? Tell me everyone. कैसा था? Physics जैसा था या अच्छा था? Was it like physics or was it event good? अच्छा, it was okayish. So it was like physics. It's either like physics or it's either gone good. It was moderate. Why this question, Shreya? It was a cakewalk for Jaya. Very good, not bad. Excellent. So kids, if physics went this good as I can see on your chat box, then bio is gonna go awesome. So yes, you are absolutely right. The paper is going to be of a 2-hour duration. Okay, 2-hour duration. This is not MCQ format. It's going to be a subjective format. All the questions are absolutely compulsory. All your questions, you have to attempt all of them. Okay, so don't start looking for अरे इसमें option दिया है क्या? No, it's not like that. Maximum option can be given inside a question, where maybe if question 4 is there, then in 4 either you have A or you can solve B. Either it can be like that. So there may be options within the question, otherwise forget it. नहीं है. Alright. Then there are 3 sections. 3 sections. You've got section A, B and C. Total are 13 questions, where all the questions are compulsory. फिर से याद दिला रहा है वो. Okay. Then we have section A is having 6 questions, 2 mark each. Okay. 6 question, 2 mark each. चलो maths लगा लो. How many marks? Then section B, you've got 6 questions, 3 marks. Okay. And finally, section C, you've got a case-based 1 question, which is going to hold 5 marks. Okay. Alright. There is no overall choice, please remember. But as I told you, internal choices have been provided in some questions. Okay. You have to attempt either one. If this kind of thing is given, then either one you have to attempt. Wherever required, draw label diagrams. If you're going to draw a diagram and not label it, you will get one big anda. Okay, no weightage without diagram. Whenever you draw a diagram, please remember that write down what diagram you have drawn. Many kids forget this. They draw the diagram, they just come out, they forget to write what have we drawn. What have we drawn? Please write that out. Okay, please write. Yes, 35 marks. So make sure that also the diagram that you draw, you don't want it to be published in the newspaper, you don't want it to be published in the textbook. Okay, please don't spend too much time in making it so beautiful. Just make sure that it is understandable, it is correct, and it is correctly labeled. Okay, please make sure about all of these things. Am I clear? Wherever you see that, wherever you understand that, oh, this has a diagram to it, draw it. Yes, absolutely draw it. But make sure you're not spending too much time on the diagram, especially if it is a 2-mark question. If you have already sufficient points, tell me if it is a 2-mark question, supposedly 2-mark questions, okay, how many points would you actually try to include? 2 marks. 2-mark questions. How many points will you try to include minimum? Minimum how many points? Always try it that way. Try it if it is possible. Please try that if it is 2 marks, you will include 4 points. Also, please make sure that your answers are in complete sentences. Please try to make your answers in complete sentences. Okay, complete sentences, and also make it point wise. Okay, point wise. That always impresses the examiner. While writing the points, please make sure sometimes you might the paper might be lengthy and you don't get time to go back and underline the important points. Sometimes that happens. So what will you do? Whatever you will write, let's say you are writing barrier. Okay, you are writing barrier. You know that barrier, in this whole sentence, barrier is an important word. So you will then and there underline barrier with your pen. Do not think that I will take a scale, I will take a pencil, I will go back and I will underline the word. वो कभी नहीं होने वाला. It's not happening at all. Okay, then and there while writing your sentence, you will underline the important points and go ahead. Okay, so please make sure that your answers are in pointer forms. Yes, let's start. Okay. Now we start with a question from section A. Okay, section A means 2 marks. So let's see what the question is. I hope everyone is able to see the screen properly. Perfect. Okay, so we see here, given here below is the picture of a transgenic crop. Okay, this is the crop picture that they have given, and we all know it is nothing but BT cotton. Okay, this is an insect resistant variety. And in these plants, a gene is incorporated which produces a toxic protein. Okay, some sort of gene has been put which is able to produce a protein which has the ability to kill the insect. Okay, what is the name of that protein and where does it come from? Okay, name of the protein, source organism. Your questions. 1 question, the source organism. 2nd question. 3rd question, what type of changes occur in the gut of the insect on consuming this protein? So 3 questions have been asked in this one answer. Okay, 3 questions. So let's see. Tell me. Yes, tell me what is the name of the protein. What is the name of the protein? What is the bacteria? Let's see over here. Now over here, in this, what they have done is they have given in a whole paragraph. I don't want this to be done in your in your answer paper. Please make sure you're going to write your answers point wise. So the first thing, the protein is called as cry protein. Okay, cry protein, and yes, and that is lethal to the insect. So that is behaving as an insecticide. All right. The protein, kids, you would write it as capital CRY, I believe. Okay, and this protein is actually formed in an inactive form called as a protoxin. That protoxin is inactive. Where does it become activated? Where does it become activated? In the alkaline pH gut. In the alkaline pH gut, it becomes activated. Now that activated toxin is going to bind to the surface of the midgut. Okay, surface of what? Surface of midgut. Please remember, surface of midgut, and that too to the epithelial cells. Then what will it do? It makes holes. It makes holes, and it's causing perforation. Don't write holes. Use nice words. Let the examiner know you know a little bit of English. So it causes perforation, causes swelling, and then ultimately lysis of the cells. So where are your what are the first which protein, which is the name the toxic protein, source organism? Okay, toxic protein is the cry protein. The cry protein. So one answer done. Source organism, Bacillus thuringiensis. Second answer done. And how is it working? Third answer done. Please make sure that even the sub questions are all answered. Sometimes what happens is when you sit to answer this, you will forget these two. Please make sure you identify how many things have been asked from you. Okay? When will I send this PDF? Immediately after this session is done. Okay. Chalo. Everyone ready? Question number 2. Acha, after this now we have a week quiz. So be with me till the end of the session. The very same chapters I have attached a week quiz. We will be doing those together. Okay. So stay with me. Now, question number 2. Question number 2. What is this? This right here, the diagram is of gel electrophoresis. Let's see what the question is. Following picture shows the apparatus involved in the process of gel electrophoresis. In the technique of this, what material, here's your question, which material is used as matrix? Also, they are asking its role. So, 2 questions have been asked. What is the material used? What is the material used in the matrix? Everyone, who is telling me? Very good. Yes, Saksham, you will join too. It is agarose gel. Very good. So, agarose, where does agarose come from? Who will tell me that? Kahan se aaya? Very good, Shreya. Agarose gel, which is obtained from seaweeds. Very good, Shreya. So, agarose, it is the most commonly used. Okay, you can go ahead and, as Shreya has written in the chat box, you can mention that it is obtained from seaweed. Okay, humko matlab, you know, this is a very common human tendency. When we know something, na, hum aisa chilla ke we tell everybody, I know this, I know this, I know this. So the same way, you know, when you learn something on our Wave platform, okay, all my kids tell me this many times. Ma'am, we learnt it in the Wave platform. Then in school when they taught us, na, I was the only one answering. So that's when you like doing that even more, na. Humko aata hai, mujhe aata hai. So वैसे you can go ahead and write down over there along with agarose that agarose is obtained from seaweed. Alright, and that is the most commonly used matrix. The basis of gel electrophoresis is that what happens is this matrix, the whole consistency of the matrix is like sieve. Okay, channi jaisa, like a channi. Okay, it's like a filter. So basically in that filter, the smaller, the lighter, the smaller DNA particles will be able to move fast because of the electric current. Okay, so how are we going to write that in words? Ma'am, we know it, but how do we write it? So look, the DNA fragments separate according to their size. Why are they able to be separated? First of all, there is a electric current provided. Second, because there is a sieving effect provided by the agarose gel. Very important. Agarose gel provides a sieving effect. Okay, and therefore the smaller the fragment size, fatafat it's gonna move, farther it's gonna move. Okay, the smaller it is, the more active it will be, the farther it will move towards the which terminal? Towards which terminal? Who's gonna tell me? Towards which terminal? Batao. Very good. Towards anode. DNA is negatively charged. Shreya, I'm quite impressed. Very good. Next question. Question number 3. List any 2. Please remember, I don't want any of you coming out of the question paper. Oh no, ma'am, I only wrote 1. Oh no, ma'am, I didn't see that sub question. वो नहीं चलेगा. So when you're reading the question only, if you want to, you do it with a pencil like how we're doing it together right now. Please make sure you read the question properly. How many advantages have they asked you for? 2 advantages. Of what? Of using a biopesticide. Advantages of using a biopesticide. कौन phone करता है बीच बीच में? Okay. So, advantages of using a biopesticide are, first, with the use of the biopesticide, there is a great reduction in the use of agrochemicals. Agrochemicals means those insecticide, pesticide, all the chemicals. Okay, so you're replacing the chemicals with biological agents. Okay, these chemicals which are there, agrochemicals, are responsible for pollution. So, by using a biopesticide, you will be able to reduce pollution. Okay, that's why biopesticides are eco-friendly. है ना? Then, second point, they want 2 advantages. They serve as a permanent control of pests. Okay, by having a long persisting effect. So what happens is these agrochemicals, now you have to keep on spraying. But these biopesticides, whenever they are used, they've got a much longer effect. So obviously, not only the environment, but even the pocket of the farmer is going to be saved. He doesn't have to keep on investing in those chemicals because they have a long persisting effect. Also, third, they have gone ahead and mentioned, क्योंकि इस बंदे को आता था तो show off कर रहा है, they leave no toxic residue. Even better. Network check. Why is it, am I audible and visible to everyone? Is there a lag? Shreya, Nivel, is there a lag? James Berg, where were you from the past 2 days? I saw you on the first day in biotechnology. Where were you? Is there a lag? There was a little. Now is it okay? Guys? Is it okay now? Thank you, Nivel. Alright. Okay, fine. Now, next question. Question number 4. How the limitation of traditional hybridization process was overcome by use of technique of genetic engineering. So only one question they have asked you. What have they asked you? How is the limitation of traditional hybridization? Traditional hybridization had its own limitations. How were we able to overcome those limitations by using genetic engineering? Any points in this? Tell me. What will you write in this question? What will you write? The traditional hybridization had limitations. First, you'll have to think, what were the limitations of traditional hybridization? Tell me. What was the limitations of traditional hybridization? Think about it. And then, how did biotechnology help to overcome them? No variations, okay? Don't worry, Saksham. No variations, okay. Anyone else? Shreya, Nivel, everyone else. What else can you think were the limitations of traditional hybridization? Yes, very good. When we did traditional hybridization, okay, there was always a very long and tedious method which was called as selection. Yes, a very long and tedious method which was called as selection. Even when Mendel was working, there was always one that was, recessive. All different varieties will come in the hybrids. But then from those different varieties, we had to actually go and select the one which we want. And we have to be very careful in those selections. Because what if the one that we want, by mistakenly you throw it off? Okay, so that was the biggest limitation that we had seen during traditional hybridization. But when we talk about genetic engineering, what happens is we know what we want. We know that that's the only thing we're growing. So no hassles of selecting the superior progeny. Okay, very good. So the limitations. Okay, The limitations of traditional hybridization process was overcome by the use of techniques of genetic engineering. What did genetic engineering do to overcome that? We created and we used a recombinant DNA. Okay, they created and they used the recombinant, and then after the recombinant was made, gene cloning and gene transfer was done. So with the help of gene cloning and gene transfer, okay, they were able to overcome all those limitations. And we know the biggest limitations was the presence of the undesirable varieties. Okay, so this is also allowing isolation and introduction of only one or a set of desirable genes without introducing undesirable genes. Excellent, everyone. Without introducing, yes, CBSE exams, last exam, biology is left. Alright, next question number 5. What is the ill effect of excessive use of antibiotics? Ill effect of excessive use of antibiotics. And who coined the term antibiotics? So, one question, second question. They've already numbered it for you. They made your life simple so that you won't skip a question. Now, excessive antibiotics will lead to what? Resistance. Excessive antibiotics will lead to resistance. The bacteria becomes resistant to it. So, excessive use of antibiotics. There are some people, cheek eye, a little cough hua, and they start taking their antibiotics. Arey, do you even know whether it is bacterial or viral? Now, even if you have just a viral infection and if you take antibiotics, it's going to do nothing. You're giving a virus an antibiotic. The virus will be like, de do, aane do, I don't care. It's not gonna affect me. So, excessive use of antibiotics can lead to infection or even colonization with the resistant pathogens. So, if at all there are people who consume unnecessarily, that too even for viral infections, it is common, and thus it leads to increasing rate of antibiotic resistance. Okay, that too, the most common examples given to you are among Streptococcus pneumoniae, okay, and even other commonly infecting pathogens. So basically the most important thing seen here is it is increasing rate of antibiotic resistance. Most important point of this answer. Okay, the term antibiotics was given by, they have not asked you that who invented the first antibiotic. Okay, all of you have given me the answer, Alexander Fleming. But over here, the term was given by Selman Waksman. Term was given by Selman Waksman. Please understand this. No, Alexander Fleming was the one who found it, but he did not. Okay. Alright. Alright. So let's move on to the next question. Name a drug. Okay, James will see that out. Okay. Name a drug used for helping patients cope up with mental illnesses like depression. So if you want a drug to cope up with depression, your drug will be a stimulant or a depressant? Your drug will be a stimulant or a depressant? Tell me. What, Ramesh? Is autoimmunity also an answer? Not for our body. The bacteria becomes resistant. Yeah, kya hai? Mixed answers. The person is suffering from depression. He is already feeling low. You want to give him, are we going to give him medicines which will make him feel even more low? You will give him a stimulant. Correct? A drug is there which helps patients cope up with depression, but it is misused. Which type of drug is this? Also, label the plant source of the drug which is popularly called as smack. Tell me. You tell me here only on this slide. Which drug is used for depression? We did this just 2 days back. Or yesterday. Yesterday only we did this. Which is the drug used for depression but it is misused? Think about it. This is heroin. Which drug is used as a depressant? So, yes, plant source, yes. So the answer here, heroin. Heroin is obtained from Papaver somniferum, poppy plant. Acha, which part of Papaver somniferum? Which part of Papaver somniferum do you remember? The source is Papaver somniferum, but which part of it is used? Which part of it? Yes, these are opioids. Very good. Which part? Very good. The latex, the sap, that the latex part of it is used. Excellent. The latex part is used. Excellent, kids. So look, these are one of those answers where only 2 points are there as the answer, so it's okay. Next we come to section B. Okay, section B means how many marks per question? Section B means how many marks per question? How many marks per question? Yes, even amphetamines, barbiturates, benzodiazepines can be used. Yes, this over here along with heroin to treat illnesses like depression, even barbiturates can be used. That can be an answer too. It would be right. But even heroin is having stimulant opioids. Okay, if you see, let's go back. I'll just show you that. I'll just show you where we have written it in our notes also. Okay. Yes. Look, to treat insomnia and depression, yes, amphetamines, benzodiazepines can be used and these are misused. So this would actually be a more appropriate answer if you write this. Okay. Also, if we see here, opioids are, heroin is commonly called as smack and it is a depressant. So not heroin would not be used. So sorry kids, heroin would not be used because you would need a stimulant. So over here heroin is wrong. Sorry kids, heroin is wrong. Heroin does not behave as a stimulant. Heroin is in fact a depressant. What will be the right answer? What would be the right answer? Right answer will be barbiturates, amphetamines. Barbiturates, amphetamines. Okay, barbiturates, amphetamines. Yes, correct. But cocaine is not given as a medicine. Okay, over here they've given a drug help for the drugs. Okay, heroin, cocaine. When you go to the doctor with for that you've got depression, doctor doesn't say okay go home and have some cocaine. Okay, the doctor doesn't say that. Doctor will give medicines which have barbiturates, medicines which have amphetamines. Understood? Otherwise that doctor will be banned. उसकी degree ले लेंगे. Alright, imagine that. A person suffering from depression goes to the doctor, a psychologist and psychiatrist. Morphine is used for as a painkiller. Morphine is used as a sedative and a painkiller. Sedative and a painkiller. Look at NCRT everyone. Properly NCRT has mentioned barbiturates, amphetamines, benzodiazepines. They are normally used to treat insomnia and depression. But then they are misused. They are misused. So that's drug abuse. Okay everyone. Saksham. Hi Probal. Alright. Now section B. Chalo, let's start with section B. We are at question number 7. A lab technician runs a PCR machine for years. Okay. By simply making use of the sample and the components which are available to him. Okay. But he doesn't have all the knowledge of all the components. Okay? He has it, he's using it, he's giving the results, but he doesn't know what is being used over there. So they want you to, one, enlist the basic components of a polymerized chain reaction to make him understand the process in a basic way. They've only asked you that one question. What are the components of PCR? Everyone tell me in the chat box. Chalo, I'm waiting for your answers. I'm waiting for your answers. What are the components used in a PCR? Tell me. Arey, those are the Savita, those are the methods. They are asking for the components. Denature, annealing and extension. So this is the process. ये तो उसको भी पता है. Even he knows it. He just doesn't know what he's using. So you need what do you need? Very first thing, what do you need? Don't you need your recombinant DNA? चाहिए कि नहीं? Don't you need your recombinant DNA first? First you need your recombinant DNA. Then you're going to be heating it. Okay, for heating it you need, and for separating it you need endonuclease. Which endonuclease? Very good, Probal. You will use Taq polymerase. Excellent. So, the basic components, first of all, you need the DNA template. Okay, the double-stranded DNA which needs to be amplified. Second, you need primers. Okay, DNA template, then primer is required. But which primer? The primers here are small length. So you're calling them as oligonucleotides. Okay, so they are short segments of DNA. Olig means what? These are primers with few nucleotides. Okay, few nucleotides. Alright, oligonucleotides. And next, we need a thermostable restriction endonuclease. We are going to be using Taq polymerase. It is isolated from Thermus aquaticus. So kids, do you see that? Even if you know the components, they have still gone ahead and they have explained the components. Sif components ka list, no, don't write the list and come back home. Okay, I told you, show off that you know the answer. You know DNA template is required, write what it is. It is a double-stranded DNA. And which DNA? Any DNA? No. The DNA which has your gene of interest. The DNA which needs to be amplified. Write it all. Okay? Write it all. Of course, Saksham. Alright? Taq polymerase. Do you know what Taq polymerase is? Yes, you do. You know it from 11th standard. Tell them that you know it. Okay? So, what is Taq polymerase? It is an enzyme. And that enzyme is obtained from where? It is obtained from thermophilic bacteria, that is Thermus aquaticus. वो भी लिख दो, where do you find Thermus aquaticus? Where do you find Thermus aquaticus? Write that also if you want to. Where would you find Thermus aquaticus? Where would you find the thermophilic bacterium? Write that also. लिख लो. Okay, and that has the property of remaining alive even at high temperatures, because denaturation is going to be done. Okay, so even though denaturation is done, heating is done, this is an enzyme which can withstand the heat and still it will continue. Okay. Everyone tell Saksham which batch I am taking. Probal, tell Saksham which batch I am taking. Okay. It is a DNA polymerase that helps in the amplification of a segment of DNA. Okay, so that is the DNA polymerase. Alright, then nucleotide bases, these are utilized or added by the DNA polymerase. Okay, so that the chain can keep on growing. So new DNA segment can be made. Okay, and you're going to amplify it. By amplifying it, how many copies are you expected to be making? Nearly 1 billion. Volcanoes, thermal vents, very good. Volcanoes, thermal vents, excellent. Yes, Probal. A, B and F. Okay. Next question. Look here. 1 billion. Very good. Next question. A famous example of mutualism, okay, is between pollinating species of wasps with specific fig plants. Okay. What are the benefits that the female wasp derive from the fig trees from such an interaction? What are the benefits? So it is Example of mutualism. Okay, what is the benefits that the female wasp is going to derive? That's what they're asking you. Okay, also, why predators are called as prudent in nature? Very good, Sharon. Saksham, it's all parallel. Molecular basis is going on. Very good. So, here is your answer. Here's your answer. We see here the female wasp is benefited by using the fruit as an oviposition. What is an oviposition? The fruit is being used as an oviposition for laying her eggs. Okay, and also uses its developing seeds inside the fruit for nourishing its larvae. है ना? So for this wasp, the fruit is being useful in two ways. One, it's going to be using it to lay her eggs, oviposition, and second, the seeds which are there inside the fig, it's going to help it nourish the larvae which will be born. And in return, what does she get? Mutualism, na? Tera bhi, mera bhi. Your win, my win, both win-win. So in return, what does the wasp give? It is going to pollinate the fig flowers, inflorescence, okay, while searching for suitable egg laying sites. So meanwhile, when she is finding her oviposition, she doesn't realize, but she is pollinating it. Okay, safe place for her eggs. Okay, next, there is a possibility of prey becoming extinct. Okay, if a predator is too efficient, if the predator is too good, not too good, so it's helping, it is going and it is doing something, but it is too good and it is going to over exploit the prey, and consequently what happens? Predator becomes extinct. ये हमको नहीं चाहिए, है ना? Too much of anything is bad. So too much of efficiency is also bad, because then what will happen? There will be lack of food. This is why predators eat the prey population in a manner, खाएंगे लेकिन इतना खाएंगे कि that it won't get extinct. Pura, don't finish everything, right? Leave some behind so that it can continue, so that it can survive. Even when your mom makes curd, na, remember microbes, even when your mom makes curd, she will never let you finish the whole bowl. She'll tell you thoda rehne de, so I can make some more. Same way here, thoda rehne do, so it won't get extinct. Because if it becomes extinct, usi ka problem hoga. They're only going to face a problem. This is why predators eat the prey population in a manner that the prey continues to survive and it is not overexploited. Understood? This is the reason that predators are prudent in nature. Excellent. Very good, Shreya. Next question. What is BOD test? First tell me what is the full form of BOD. Everyone on the chat box. What is the full form of BOD? Who will tell me? Full form of BOD. Who will tell me that? Full form of BOD. Saksham. Biochemical oxygen demand. Very good, Irfan. Biological oxygen demand. Okay. Very good. Oops. Sorry. Alright, so what do you mean by BOD test? First question. Okay, when is this test performed during sewage treatment? Second question. Then we'll come back to this question. Okay, look, what is BOD test? First of all, BOD is biochemical oxygen demand test. Okay, it refers to the bioassay procedure. What does it do? It is going to measure the oxygen consumed by a bacteria. That's what it is all about. It will measure, most important, it measures the oxygen consumed by a bacteria. From where? How? How is it consuming the oxygen? From decomposition of organic matter over a period of 5 days at a temperature of 20 degrees. So if you have organic matter and there is bacteria inside it, the bacteria will decompose that organic matter. Okay, given that its favorable conditions are there. What are the favorable conditions? Give it 5 days and keep it at a temperature of 20 degrees. So BOD is going to be expressed in milligrams per liter of the sample. Okay, milligrams per liter. And biochemical oxygen demand, when is it used? That was the second question. Remember, what is BOD and when is it used? During sewage treatment. So during sewage treatment, it is used in secondary sewage treatment. Secondary sewage treatment or biological sewage treatment. Okay, they are going to wait until the biochemical oxygen demand goes low. Okay, so when the BOD of a water is low, what does this indicate? It indicates that the organic matter has been decomposed. Okay, it indicates that the organic matter has been decomposed. So that's when they know that the work of the bacteria is done, the water is now clean and it can be taken for the next step. So let us now come, we're done with question A, let's move on to B. Okay, 3 samples of water are taken. Their BOD was measured to and found to be 40, remember it was measured as milligrams per liter. The first one was 40 milligrams per liter, second one was 10 milligrams per liter, and the third one was 600 milligrams per liter. Which sample is most polluted? Remember, when BOD is high, that means that the organic matter is high, and organic matter is more means it is more polluted. Yes, so we see that BOD level and oxygen content are inversely. So kids, you would want to write this description down. You would want to write this description down because remember, this is section B. So you can go ahead, you can explain what is BOD, what is the relationship between BOD level and oxygen level. They are inversely related to each other. Thus, when the BOD levels are high, okay, then the dissolved oxygen levels decrease. So you can say in other words here that BOD levels are inversely proportional to the oxygen consumption content, oxygen content. Okay, so correct, because the oxygen present in the water will be consumed by the bacteria. Therefore, higher the BOD, more the water is polluted. So the one with 600 milligrams per liter, that is option C, the third one, is the highly polluted water. That is the water which was highly polluted. Clear? Question number 10. How biofertilizers are responsible for increasing the fertility of soil? How are biofertilizers going to help to increase the fertility of the soil? Let's see here. Okay, any points from your side? Tell me. Any points from your side? Please tell me. Yes? Rakesh, Meha, Sumit, tell me any points from your side. How are biofertilizers responsible for increasing the fertility? Very good. Nitrogen fixation. Excellent. So, a biofertilizer can be defined as a fertilizer that's made up of living microorganisms. What does it do? It promotes plant growth when it is applied to the field. How? It increases the supply or availability of primary nutrients. Therefore, can you see kids, you gave me an answer in one sentence. You gave me increases nitrogen fixation. But look at how they have come to the answer. First, they've told you that biofertilizers promote plant growth when it is applied to the field. Okay, alright, so this is where you're not going to be answered that dot on. You will kind of thoda khichdi banao, hai na? When you don't tell them that you just done tadka. Tell them you put oil, you put jeera, you put hing. Tell them all of that. Okay, give them the details. Only then you'll get the full marks. So, it promotes plant growth when applied to the field, increases the supply of the primary nutrients to the host plant, right? Therefore, it helps in increasing the fertility. Main sources are several species of bacteria and cyanobacteria. What do? They have the ability to fix free atmospheric nitrogen into organic compounds, and that's how it can be made available to plants. Give me examples of this. Give me examples of this. Examples of bacteria and blue-green algae, cyanobacteria, which are doing this. fixing free atmospheric nitrogen. Who knows the examples? Tell me. Very good, Irfan. Rhizobium, excellent. Any others? Any others? Because this can be just an add-on question and state examples. Nostoc, Anabina nostoc, excellent. Anabina azola, but Anabina azola is what? Mycorrhiza to comes in fungus. Mycorrhiza will come as fungus. And mycorrhiza increases it in what way? By increasing the water content. Water content. Okay. Excellent. Question 11. Question 11. Enlist the benefits that are demonstrated by plants with a mycorrhizal association. Abhi discuss kiya. Okay. So plants with mycorrhizal association demonstrate the benefits like resistance to root-borne pathogens. Okay, tolerance to salinity and drought. Okay, and an overall increase in the plant growth and development. Okay, tell me which are the two types of mycorrhiza association. The two types of mycorrhizal association. One is forming. Okay, they are able to tolerate abiotic stress. Very good. They're able to tolerate abiotic stress. Excellent. Symbiotic relationships. Okay. Alright. Now, are you ready? Shall we go for the next answer? We come to now section C. Section C, question number 12, only one question, case study question. Let's read this together. Alien species or non-native species, alien species, okay? They are often introduced deliberately into a habitat for their economic and other uses, okay? They become invasive and they drive away, they become invasive. Like how you saw cancer cells, they become invasive and they will drive away local species. Okay, now exotic species have proved harmful for aquatic as well as terrestrial ecosystem. Okay, the following picture, what picture? Look at this picture. Okay, the following picture shows an invasive plant. Okay, that is water hyacinth. Alright, and that was introduced in India because of its beautiful flowers and shapes of leaves. But was it good? No. What did it do? It clogged many water bodies and that resulted in death of aquatic plants and animals. Now, based on this, answer the following questions. Okay, this had happened in your term 1 exams also, and we all always wondered कि भाई इतना सारा क्यों लिखते हो? Just give the questions, we'll give you the answers. But let's see what they're telling you. You studied this in botany, very good. Define invasive species. First question, define invasive species. So, what is invasive species? The alien species that is unintentionally or it is deliberately introduced for different purposes. Okay, for different purposes. They will turn invasive, they will cause decline or extinction of indigenous species. Okay, so whichever ones are deliberately put, okay, for somewhat reason, but they are causing harm to the neighboring ones which are already there, the native plants over there. That is what we will call as a alien species or invasive species. है ना? These are those people who come and stay in your home, don't pay rent and don't even talk about going. ये वो इंसान, ये वो organisms है. They come to your house, they don't pay you rent, they are just a headache for you. Bin bulaye, they've come and they're not talking about going also. Yes, we all have them in our life. Then, name one invasive species introduced in India apart from water hyacinth. Very smart. They know you will take this and write that only. Upar ito likha hai. This is not English comprehension. This is not English comprehension. So very smartly they have told you. Give another example. So another example is going to be invasive weed species, carrot grass. Okay, Parthenium. Or Lantana, let me zoom this in. Lantana camera was introduced in India that led to environmental damage. This is how it has proven to be invasive. It caused damage to the environment and it posed as a threat to our native species. Okay, next, what do you mean by endemic species? What are endemic species? They are, endemic species are a species which is native to where it is found. है ना, घर की मुर्गी है ना. So next, mention any two ways in which invasive species affects an ecosystem. How will it affect an ecosystem? So many ways you can find out. Okay, remember one thing, kids. Please make sure that if it's A, then in your answer you've written A. Mark 1. Please make sure what sub question they have given in what form, and that is exactly how you will write the answer. So now this is B. B ke jagah pe don't write 4. If I am your examiner, you'll get a big fat anda. Okay, because we have literally got 000 marks because of this when I was in school. So yes, now we come to B. Invasive species can be a major cause of biodiversity loss in the ecosystem. Okay, what do? They come and compete with native species. हमारे native species are all bichara poor native species, and the other ones come and they compete. What do they compete for? For resource. Okay, different types of resources they will come and compete for, and therefore the native one becomes extinct. Okay, that can reduce biodiversity. I'll give you an example. There are those people with the local shops, in the market, sabzi wale, everything is there. They all are working very hard. They're doing everything. But then what happens? In comes all these online shopping, or you can say, in comes the ones who come and deliver the food at your home. So now obviously you're getting delivery at home, but what is happening to the people sitting in the market? It's affecting them. Same way, another species is being introduced, it's hampering the native species. So the invasive species will cause extinction, and that is going to then also, why do you want to prevent that? It will alter the food web and its habitats. Clear everyone? Now are you all ready for the quiz? Everyone ready for the quiz? Everyone ready for the quiz? Are you all there? Now, everyone, I hope that you have gone and liked the video. This, if you've not done so far, this is the perfect time to do so. And also, in the description box, you can find the link of the V quiz. Jaldi se aa jao in the V quiz. Everybody quickly come to the V quiz. I am already there. Quickly come to the V quiz. I am already there. Come to the V quiz quickly. I am already there. Okay. Please put in the code 225009. 2 people have come so far. Quickly everyone. Where are you guys? 225009. Come quickly everyone. How many people? 3 people have come only. In the description box, the link is there. Click on the link, put the code 225009 and join with me. Don't be scared, Saksham. Just come. Can you find that link in the description box, everyone? V quiz link. Join that link. 6 of you have come so far. Where are the remaining 20? Join the link and put the code 225009. I will not start until all of you come over there. Quickly, I am waiting for all of you. Come on. Only 9 people have joined so far from my chat box. Join the quiz link. There's a V quiz link in the description box. Everyone, quickly, I am waiting for you. 10 people have joined. 27 people are there right now. Kids, join quickly the description box, the link there. Ramesh, can you see the description box? Can you see in below the video is written biology sample paper? Please click on that. There is a link written there. It's called V-Quiz link. Did you find it, Ramesh? It's okay. Even if you didn't ask for code, maybe it just let you in with the link only. It's fine. If it asks you for the code, then put the code. If it doesn't ask you for the code, then maybe you've just joined in. So don't worry. Here you go. Thank you so much. Here is the link. Thank you so much, Enlite ji. So we've got the link over there. If the code is asked, hi Tashmeen. Tashmeen, quickly, you've come at the right time. Please join the link so that you can play the quiz with us. How many of you are here? 25 are on my chat box and only 12 are in the quiz. Why? Now though we've got the quiz link on the chat box only. You just click on that link. Click on the link in the chat box, everyone. 13 people are there in the quiz. I won't start unless 17 people come. Hi Anishka. Alright, so shall we start? Okay, 14 people have started and let's begin. So, your first question on your screen, answer fast to get more points. In gel electrophoresis, separated DNA fragments can be visualized with the help of... Okay, Ramesh, answer the questions. And time's up. Okay. So yes, ethidium bromide in UV radiation is the right answer. What is the color that will come? It will be orange color. Alright. So let's see the leaderboard, and we have Muthu Minakshi at first place, Charisma, then Adi. Then comes Aryan, Anshika, Srijani and Shreya, Irfan, Surya, Ilanthrendal. Wow. Next question. A specific recognition sequence identified by endonucleases to make cuts at specific positions within the DNA is... What is the recognition sequence and what is it called as? All of you who are there who have not joined the chat box, please join so quickly. Acha, let me, so sorry, I guess you're not able to see it properly. Option D is not visible. Now is option D visible to everyone? Very good. It is the palindromic nucleotide sequence. Very good. Palindromic nucleotide sequence. Now, leaderboard. And yes, Muthu Minakshi is still at first place. Aryan and Anshika. Excellent. Shreya, buck up. Next question. Which of the following is the most important cause for animals and plants being driven to extinction? Animals and plants being driven to extinction. Meha, please try again. Everyone has joined, beta. The link is there in the chat box also, in the description box also. Please join again. Time's up. And everyone has... Most of you have gone for A, which is the right answer. That is habitat loss and fragmentation. Leaderboard pay. Now, Muthu Minakshi is not moving from her place. Excellent. Anshika and Aryan also decide to stay there only. So kids, come on, let's see some changes in the leaderboard. Meha, were you able to join? When gene targeting involving gene amplification is attempted in an individual's tissues to treat disease, what is it called as? To treat disease. What is it called as? And time's up. Yes, the correct answer is absolutely D, that is gene therapy. Excellent. You want to treat disease, now so therapy naturally comes there. Oh, Aryan has come to first place. Muthu Minakshi putting her at second, and Adi is at third. Very good. Next question, guys. Answer fast. DNA precipitation out of a mixture of biomolecules can be achieved by treatment with. This is something everybody should be knowing. Why not working, Mihika? I've already have 16 people there. Please try again, beta. Why is it not working? So sorry. Okay. Yes, the correct answer here is going to be chilled ethanol. Excellent. Correct answer is chilled ethanol. Sorry, kids. There was a small error from my end. And yes, the correct answer is chilled ethanol. Let's see the leaderboard. Very good. Aryan, Muthu Minakshi, Adi is still at third. Excellent. Next question. Try refreshing, Meha, and join back. What is the criterion for DNA fragments movement on agarose gel during gel electrophoresis? What is the criteria? It's okay, Saksham. Very good, Mehika. Excellent. Aja, aja. So yes, the smaller the fragment size, the farther it's going to move, because the agarose gel is providing a sieve, and adding an electric current will make it move. The negatively charged particles go towards anode. Very good, Muthu Minakshi. Adi and Srijani upar aa gayi hai. Shreya has also come up. Aryan, what happened, Aryan? Aryan, these girls are beating you. There's Adi also beating you. Alright, let's start next question. Answer quickly. What is the competition between individuals of two separate species for sharing the same resources in the same area known? Oh yes, people are laughing at my home now. Yeah, my son is having a friend over. Apparently, they become so hyper when they are near each other. Oh my god. Yes, and the correct answer here is interspecific competition. Excellent. Interspecific competition between two species. Interspecific. Muthu Minakshi, very good. Srijani and Adi, excellent. Okay. Next question, eighth question. Answer quickly. The drug heroin is synthesized by, this is something we all should be knowing. Is it synthesized by methylation of morphine, acetylation of morphine, glycosylation of morphine, or nitration of morphine? Right out of your NCERT textbook. Kya baat hai, Saksham? Right out of your NCERT textbook, this question. Very good. As heroin is synthesized by acetylation of morphine. This is a previous year question. And Muthu Minakshi stays at first place. She was at second for a little while, but then she came back to first. She's like, I'm not moving from here. Good job, Srijani and Adi. Shreya, I want you to go even faster. Next question. Which body of the government of India regulates GM research and safety of introducing genetically modified organisms for public services? And you have 9, 8, 7 and 5 seconds left. Okay, and time's up. So yes, that's absolutely right. As I tell you everyone, that the scientists are going to go bizarre, but yes, we're gonna have engineers who are going to come and tell the scientists whether it's approved or not approved. At the end, the engineers are gonna come and tell these scientists what to do, what not to do. So it is the Genetic Engineering Approval Committee. Let's see your leaderboard. Excellent, Muthu Minakshi. You have outstanded the performance. Very good. Shri Jani and Adi also excellent. Very good, Mihika. Tobacco plants resistant to a nematode have been developed by the introduction of DNA that produces what in the host cell? What is the DNA produced in the host cell? Very nice question. Meha, Meha, not on the chat box. And yes, it is going to produce both a sense and antisense RNA, which is a double stranded RNA, which is going to be silenced in your eukaryotic cell. Excellent. So, leaderboard. This is the final leaderboard. Yes, and good job, Shrijani. We have Adi at second place and Anshika at third place. Shrijani just jumped. Shrijani just jumped. Kaise? So, Adi at second place, Anshika at third place. Very good, Shrijani. Absolutely wonderful. I hope that you guys enjoyed these quiz. I hope you enjoyed it. Let's just come back. Yes. So, where were we? Yes. So, kids, just so that you know, CBSE Term 2 is not everything. Okay, your NEET exam is the main goal that you all have here. So, yes, for all of you, Vedantu has introduced a very lovely way to make sure that the next 50 days of yours are spent perfectly in the right direction. We have the NEET Crash Course Lite. The course is including 120 recorded interactive sessions. Yes, absolutely. At Vedantu, everything is interactive. Okay, even if it is a recorded session, so that it fits your budget, it is going to be interactive. There is no such thing as no interactive sessions. So it is absolutely interactive. We are going to have a classroom there. Your friends will be there at the very same time. And the best part about it is that every doubt of yours while you're watching the recording, you know this happens now. Every time, even if you watch any recording, there will always be doubts, right? Also over here in the sessions also now, if you have any doubts, what I would want you to do is after the session or when you're watching the session, let's say a replay or something, please in the comment section I would want you to post your doubts, so that whatever doubts are there, I will be able to solve it in the comment section itself. Okay, maybe I won't be able to solve your doubts during the live class right now, when I'm taking it live, but whenever you feel so, you can post your comment in the comment section there. We can take care of it. If there's any topics that you want me to take up, if it's feasible, please post it there, so we know how you would want the upcoming sessions to be. Okay, we will always be able to consider those if it is possible. Also, I'm coming up with sessions for only diagrams for 11th and 12th, where we're going to we're going to be going through all the different important diagrams which are there in 11th and 12th. Also, as promised to all of you, I'm keeping a session for earthworm. I think on the 30th of this month we're having earthworm session. So yes, if you just let me know, we can conduct all those sessions. A live doubt support in the class with the class teachers will always be provided, and there will be 10 part tests and 10 full tests until your NEET exams. All of these recordings will be provided to you, and you will have access to them until the day of your NEET exam. There will also be with every lecture there will be included assignment. So total number of 90 assignments to make sure that each and every concept is covered with MCQs. Alright, so let's see who are these brilliant teachers that are going to be taking these crash course recorded sessions which are there. Yes, we've got Rahul sir for physics, an awesome sir to help you out with the basics which might be weak, with the things, most important parts which need to be done just before the exam. Then we have Sudarshan sir for chemistry and our very lovely Bhumika ma'am for biology. Okay, excellent, awesome teachers, I'll tell you. Here is the physics schedule. You can see that all the topics are going to be covered as per required, that too right from scratch. chemistry schedule and botany schedule. You can see here in botany also everything is covered. All the chapters of 11th and 12th standard are going to be covered, as well as the zoology schedule. So you can see here, you can take a screenshot of these kids, and the best part about this is when these schedules are going on, when these courses lectures are going on, you will have the leverage, you can say, from any other crash course available in the market. You will have the leverage that you can always unmute yourself, you can ask the teacher to uncam you, and you can have a live interaction session with the, or any, you can have any doubt solving with the class teachers right there. Okay, what is it that the Vedantu crash course is offering that other crash courses are not? Yes, class interaction is very high, others are not. Okay, in class doubt support, quizzes will be there. You know how fun these quizzes are. Hotspots will also be there, and live polls are there. And there is always going to be milestone, that is a learning outcome. Okay, after every learning outcome you will know that today I have achieved this much. And at the ending, always what you guys wait for, the leaderboard will always be displayed. How much is this for? For the next 50 days, to make sure that your the direction in which you're approaching the final days of your NEET exam is in a very perfect way. Only and only 2700 after discount. And the recordings, as I told you, will be available until your NEET exams. If you still have any queries or any questions about this crash course, please feel free to WhatsApp your queries at this number. Take a screenshot of this, and you just need to enter what your name is, which standard you're in, and just mention that you've texted after watching my session. Okay, please note down this number. And yes, the crash course is available now. Okay, you can get it at a discount of 2700, and just by putting a coupon code SIPRO, you get a great deal. The link for the crash course, kids, it's available in the description box, which is mentioned right where you saw the link for V-Quiz. Just go a little below. You will see the for the crash course link is also there. Please don't forget to apply the coupon code so you get your discount. All right. And yes, I will see you all tomorrow. I'll see you all tomorrow. We're discussing one more sample paper just like we've done today. Alright, so everyone be there. Yes, Mihika had fun too. Perfect. So tomorrow also will be same way. We will be having a sample paper, and after that we will be having a quiz round. Alright. All the best, you guys. I will see you tomorrow at the same time. Bye-bye. Ramesh, we will be doing organisms next month in June. Okay.